All right, we're finally going to get into metabolism and the biochemical processing of different types of food. So this first part's just really an overview, a big overview of what happens to the various components. And the general idea is that we have these three major food groups that we've been talking about: lipids, carbohydrates, and proteins. They get processed into fatty acids, and they all kind of, in order to generate energy, right, they can be made into other things. We'll talk about that as well. But in order for these to make energy, they've got to go from these larger grouped compounds into smaller compounds that are eventually going to turn into carbon dioxide and water. So these larger chains of carbon are going to eventually get combusted into CO2 and water. And there are a lot of different types of enzymes that are involved in the process, from lipids, carbohydrates, and proteins down to those smaller compounds, those molecules. So the first step, we've already talked about that digestion, right? Where the saliva, we have different enzymes in our saliva that start breaking down some of these compounds. We have ultimately carbohydrates are going to be broken down into glucose, proteins are going to get broken down into amino acids, and lipids are going to get turned into fatty acids and glycerol. So we've already kind of talked about that in the digestive stage. So we've gone through those. And these smaller molecules get into the enterocytes in our small intestine, and they're brought into the cells and then pumped through the blood so that they can be part of our bloodstream, and we can then our cells can access those nutrients for metabolic needs. So let's start with glucose. This is kind of the sugar. Any carbohydrates are going to eventually get converted into glucose that's going to be used in our cells. So glucose is brought into the cells, and we'll talk about this in a little more detail. But one of the first things that happens when a glucose comes into a cell is it gets phosphorylated. We have this glucose 6-phosphate, and that actually traps it into the cell. Remember, we talked about these things being able to kind of go. There's a shuttle protein that brings in glucose, and glucose can go in and out of that membrane protein, except for once it's phosphorylated, it gets trapped inside. Now that membrane-bound protein doesn't recognize glucose 6-phosphate as a substrate for it, so it gets trapped inside the cell. So this is one of the first things that happens. And as you can see, we kind of add more phosphate groups, we kind of move some stuff around. We break this 6-carbon group into 2 3-carbon groups, and those little smaller chunks of carbon groups are getting reduced and oxidized. We generate some energy, and then we ultimately get down to pyruvate. But ultimately, this all comes down to pyruvate. We get some ATP out, as you can kind of see in this step. In this step, we also use some ATP up at the top end as well. But we do get some overall net ATP out, and we also get pyruvate. And pyruvate's a major intermediate. Pyruvate is a compound that you should get to know. We'll come back to this many times. And the final step here in glycolysis is pyruvate and ATP is generated. So phosphoenol pyruvate is this compound over here. There's your pen. Phosphoenol pyruvate. You lose that phospho part, and then we generate just the pyruvate. Notice the enzyme name. This is a kinase. That means kinases are phosphorylating or dephosphorylating compounds, and pyruvate is the compound that it's acting on. So actually, you'll notice that the chemical reaction is going this way. Sometimes we'd say a dephosphorylase, but right, the compound it's actually interacting with is the pyruvate kinase. It's actually phosphorylating pyruvate. But we know chemical reactions can go both ways, and this is one of those where the chemical reaction, we actually have the name of the enzymes, actually the product in this instance, and we're dephosphorylating. But we're talking about energy here, and I mentioned earlier that we talked about phosphorylation as an important mechanism for getting chemical reactions to move forward, because it's a higher energy. We know it has energy in this bond when we go from one step to the other. But we're actually making ATP, and we're making energy, right? We're making one of these high energy bonds. But we're also breaking a bond here. So let's see how that actually works out. So these are the free energies involved in the hydrolysis of phosphoenol pyruvate and ATP separated out. So here's the phosphoenol pyruvate, and there's water. And we're going to hydrate this. We're going to actually break this bond, and we're going to form pyruvate. So we get this movement of a double bond up to this oxygen, that we have inorganic phosphate being created. And the overall delta G is negative 14.8. Now we're also making energy, right? We're making this ATP compound, and that's going to have a positive delta G value. So it's not spontaneous. But when we couple these reactions, when we couple them, then we actually can have something that moves forward. So if these two reactions are coupled, the delta G for the total thing is what? Right, it's negative and it's favorable. Because if we add these two reactions together, then we can actually get, right, so we're going to cancel out, we're going to, on either side of those arrows, we're going to cancel out the kind of components that are shared, and we actually can get the phosphoenol pyruvate plus ADP plus the pyruvate and the ATP, and that's going to give us an overall negative delta G value. So this overall coupled reaction will be favorable. So this is the next major component, acetyl-CoA. You can see where the lipid, we're talking about carbohydrates right now, that's one of the process, but lipids also come through this acetyl-CoA metabolite. We also have some proteins, amino acid catabolism comes through acetyl-CoA. So another major component in metabolism. And we're going to start talking about acetyl coenzyme A, right? That's part of this process. This is a coenzyme, and it's a non-protein chemical helper. So it's going to be involved in NAD and FADH oxidation reduction. Here we have the acetyl group, 2 carbons and a double bonded oxygen. I'll show you in the next slide here too, just kind of the chemical structure. 2 carbons, double bonded oxygen, sulfur, and then this big long coenzyme. You see the sulfur here, that double bonded oxygen, and then the big, the CoA part of this. So I'll just show you. This is, right? This is the sulfur. This is the oxygen. These are the 2 carbons up here, and then the rest of this is the coenzyme A. These 2 carbons are important. They're actually the small molecules that are coming from lipids and proteins and carbohydrates are digested into these 2 little, all those longer chains of carbons end up on as these 2 little carbons on this long chain that are attached to the sulfur, that are attached to this longer subunit group. So the acetyl groups are attached to the coenzyme by the thiol group, and they're important intersection for a lot of different pathways. Here we can see that acetyl group again, just as in the chemical form, but then with the coenzyme A part. So we don't have the whole entire molecule in there, but just remember that that is an important coenzyme, again, a non-protein chemical helper. It's actually derived from adenosine triphosphate. And again, it's a hub of metabolism. So here you have that carbonyl group and a methyl group, and this red whole group that's actually a functional group we call an acetyl group. Those 2 carbons I just talked about, the acetyl-CoA carbons, those 2 acetyl carbons, actually get pushed into the citric acid cycle. And it's a cycle because it's a cyclical component of carbon. These carbon metabolites, we have a 4-membered chain of carbon, and those 2 acyl groups are attached to that, and we create a 6-carbon chain, and then we have 6, and then we get rid of one of those carbons as CO2. We do that again in another step, so we have 4 carbons. We rearrange that and generate some chemical energy through ATP, or we save some chemical energy, and then we kind of keep on that 4 and 4 and 4. We make some reduced coenzymes, so that means that these things are getting really oxidized, and then we add some more carbon, and then we do the whole thing over and over again. So overall, the net result of the citric acid cycle, we get 1, 2, 3, 4. We get 4 reduced coenzymes, and we also get a GTP, or an ATP equivalent energy output of this. And we also get we get carbon dioxide as well. Interestingly, ADP, so a low energy compound, ADP, not ATP, it acts as an allosteric activator of this cycle. NADH, which is, we know, a reduced coenzyme, acts as an inhibitor. ADP, that tells us that we have low energy and it activates one of the enzymes in here. NADH, that means that is a reduced coenzyme, right? That is actually an inhibitor. So if we have a lot of reduced coenzymes, we don't need to make more, right? So we're going to slow this down. So these both work as allosteric inhibitors or activators in different parts of this process to make this go faster or slow it down. Not only do we need to know what's happening with those molecules, but we need to know where they're happening in the cell. And the citric acid cycle part is happening in the mitochondria. And we're going to talk about why that's important. It's because these reduced coenzymes, that's where a lot of the energy in this whole process is generated. It's not generated throughout the glycolysis or even the GTP or the ATP equivalent that we get out of the citric acid cycle. It's actually from the reduced coenzymes going through that electron transport chain. And here is an example of lipid membranes. They're really important for the ability and the allowing for the transport of ions and creating these gradients. Remember, we talked about putting things into in organizing different things. And one of the things that we can do with our mitochondria, this is the most horrible mitochondria, right? We can make different compartments and we can organize these and we can put things in different parts of that and we can create order. So it helps us to create order and store energy. So once we have order created, we can allow chaos to form, and that also allows us to release energy. So remember that importance of compartmentalization, of order, and mitochondria are those little compartments where that magic happens. Here's what's going on with, we start with a pyruvate, actually gets transported into the mitochondria. We just talked about that as an important kind of endpoint of glycolysis. We get our CO2, we get our NADH, we get our coenzyme being bound to this kind of reduced pyruvate molecule to generate the acetyl-CoA. This all happens inside that mitochondrial cell. So once we have the acetyl-CoA, and we are going to, the net result is we oxidize the food, and that generates those reduced coenzymes. And we refer to oxidation as the loss of electrons or gain of oxygen bonding to carbon, right? Because the oxygen is more electronegative, it's going to be kind of taking some of those electrons away from the carbon technically. So the carbon doesn't fully lose the electrons, but it's not evenly shared. So we refer to that as kind of a loss of electrons with carbon and oxygen. Reduction is the gain of electrons or the gain of hydrogen as well. And that's happening on the coenzymes that are involved. And these things always happen together. So as our carbon groups that we're eating go down this chain, they're getting more and more oxygen bonds. Here we have a single oxygen bond, we have a double, we have triple, we have four, quadruple, right? And that's why we breathe out carbon dioxide, because that is the most oxidized form of carbon, and we can then we must have reduced something else to get to that point. Which structure is in the most reduced state here? So I'm not going to tell you the answer, but you should be able to figure that out based on what we just. All right, so pyruvate's been transported into the mitochondrial matrix. The electrons from the reduced coenzymes are passed from the molecule to molecule down the electron transport chain. And we'll talk about that as well, a little more detail. And along the way, their energy is harnessed to push hydrogens across the gradient, across the membrane, and they store them in that inner membrane space, and that then creation of order is used to actually synthesize ATP. That's all happening stage 4 in ATP production. We can see this is just the picture of a cell. There's a little mitochondria in the cell. Here's a blown up picture of the mitochondria. We have the mitochondrial matrix in the middle, and we have that inner membrane space here. That's where the hydrogen gets kind of increased. So there's these two membranes. So we get this hydrogen content really high in this darker brown area. And then here we have a zoom in picture of the matrix here where we have the ATP synthase. And this is where the ATP gets produced. And here I have another cartoon of that. Here's the inner membrane space. As we have these reduced coenzymes, you'll notice NADH. We have succinate. So this is a different pathway in that glycolysis citric acid cycle we'll talk about. But we get NADH, we have FADH being created, and we actually reduce these and then we oxidize them. And that process allows us to push hydrogens with their without their electrons across this gradient into the inner membrane space. And this high concentration of hydrogen allows us to actually pump the hydrogens back through this channel and harness that energy to synthesize ATP here. So again, these are the reduced coenzymes. We have complex 1 is NADH, FADH2 is complex 2, or also coenzyme. And so FADH is immediately oxidized there by the reaction with coenzyme Q here. And then, so they follow the formation of that. The mobile coenzyme Q is actually, reductions occur where electrons are transferred through that process. And so when we have this in this system, what would you say? Is the entropy positive or negative? Does this require energy or release energy? You should be able to answer that question. If you cannot, then you should go back and kind of look at some of the other PowerPoint slides and come back and figure that question out. As we go through these different complexes from complex 1 to complex 4, they contain several carriers of electrons and the hydrogen ions. They move through pathways that are completely lowering energy as they proceed down the chain here. So if we look at these chemical reactions, right, they can easily flow. It's kind of like a waterfall. Plants also contain mitochondria and chloroplasts, where chloroplasts kind of do this opposite reaction in a series of reactions that involves electron transport chains. So they do something very similar as well in the chloroplasts. So these are the coenzymes. They always have to be available. We need continuously cycle between oxidized and reduced forms. So there have to be a lot of availability for both of these. Nicotinamide adenine dinucleotide, or NADH. This is used as these are oxidizing agents and these are reducing agents. So we need these things to be available at all times so that we have these coenzymes available for any chemical reaction that might be happening at any given moment. Otherwise, that is a reactant that's not available and the chemical process will be halted. I just want to show you the exact location. So this is the chemical structure of NAD+. We have this nitrogen-bearing ring here. So this an aromatic ring with a amide functional group on the end. What we actually do is we take 2 electrons here, and these 2 electrons will interact with these 2 hydrogen ions, and we're going to actually add one of the 2 electrons into this bond. So we're going to actually take electrons from this ring here, and we're going to use them to create this new hydrogen bond. So this is the reduced version of NAD, so it's NADH and H+. So we've actually taken both of these, and this is what we're talking about when we have NADH, H+. So this second hydrogen atom removed from the surrounding oxidized substrate then enters the solution as this hydrogen ion. We also have FAD. This is an example. We have this long chain. This probably looks familiar. It should look like something like ATP. It's only got 2 phosphate groups, this long chain of kind of sugar-like compound, and then it's attached to this nitrogen bearing these rings, this aromatic structure here. And so these are actually the sites of reduction. So this is why we actually have a hydrogen being bonded to this nitrogen and that nitrogen here and here. And that's why we go from FAD to FADH2. So now we're actually bonding hydrogen here. We're getting from the oxidized form to the reduced form here. So it's important that the hydrogen atom is like a hydrogen ion, right, the H plus, with plus an electron. So the electrons, that's an important part of oxidation or reduction, the gaining or losing of an electron. And so when we add more hydrogen bonding, we're actually gaining electrons for that system. Here are this is an example of malate. Malate's going to be in this more reduced form. And then we have these hydrogens are lost, and we have a more carbon to oxygen bond. So this oxaloacetate is oxidized. And then we take that coenzyme and we are able to reduce that coenzyme while the oxaloacetate is being oxidized. ADP allosterically regulates isocitrate dehydrogenase complex, and that's this third step in the citric acid cycle. So I want you to think, is it going to be an activator or an inhibitor? And why do you think that? So you should be able to just think about what this compound is, how much energy is involved, what this whole process is, right? This is right now we're thinking big picture ideas. What is the goal of the cell at this moment? If we have a lot of ADP, do we want to be going through this process or do we not? And NADH also regulates this enzyme. Is it an activator, inhibitor? Why or why not? What reasons would it be that it would be an activator or an inhibitor? Again, I'm not going to tell you the answer to this. If you don't know, you should go back some slides and re-listen to some of the lesson. One thing I also want to just touch on is this whole process of metabolism. It requires a lot of oxygen, right? Oxygen's inside. We have a lot of oxidation, reduction happening. Normal metabolism requires oxygen, but it can also result in oxygen consuming reactions happening that are not necessarily good for our cells. So we can create different types of oxygen species, and that can influence the amount of oxygen. Once we get older, we have different injuries, different chemicals or drugs, toxins in the environment, different inflammation. And we can actually create from oxygen reactive oxygen species. I feel like this arrow should maybe go here, from oxygen to normal metabolism, but then oxygen can be made into reactive oxygen species. Those usually have free radicals or they're peroxides that can cause damage. So we can cause damage to mitochondrial cells. So the superoxide, different free radicals are strong reactive agents, and they can break covalent bonds in enzymes and other proteins. So reactive oxygen species can cleave proteins and enzymes that can cause major problems. It can break apart lipids, lipid membranes. It can make, right, which is really important for energy metabolism. It can get into our nucleus. They can break apart DNA. We can cause DNA damage. So all sorts of long-term health consequences can result in high reactive oxygen species. These are also sometimes referred to as ROS, reactive oxygen species. Common to look for these and to look at ways of reducing those. So this is why we a lot of times think about antioxidants in our foods. So we have our antioxidants that help to essentially absorb some of these reactive oxygen species and get them out of our system before they can cause damage and injury to our cells. There also are enzymes in our bodies, in our systems, that can convert harmful reactive oxygen species to non-harmful molecules. Here's an example of where we have this superoxide ion, which is a reactive oxygen species. You notice this one electron here. It's going to be a free radical here, and we have two hydrogen ions as well. And superoxide dismutase, or SOD1, is a compound that's known to actually take on these superoxide ions and generate hydrogen peroxide and water. Catalase is one of the most effective, efficient enzymes that we have in our bodies, and it can quickly take any hydrogen peroxide and convert it into water and oxygen. So this is SOD1 has actually been found to be important in ALDS, or Lou Gehrig's disease. And this system is basically, if there's a mutation in SOD1, it's not working effectively, and so it's been found to have neurological effects when this isn't taking care of our reactive oxygen species effectively. So a lot of the antioxidant vitamins, it's important to have vitamin E, vitamin C, vitamin A. They all help to bind to reactive oxygen species, and they help to mitigate some of the harmful effects of the reactive oxygen species. Here's an example, vitamin E, of doing that. So antioxidants and vitamins, this hydroxide group, vitamin E, once it's bound to one of those oxidative reactive oxygen species, it can actually be itself oxidized. So this is a product of vitamin E reacting as an antioxidant. So we get that hydroxide turns into a carbonyl group. We get another one on this side. We actually kind of break this chain. That's what's happening. We're breaking this bond here, and this oxygen becomes a hydroxide, and then we actually have a double bonded carbonyl group here. So we can actually do a lot of things to mitigate some of the bad reactive oxygen species. Other antioxidants include, you might have heard of resveratrol. Green tea has some of those. A lot of red wine, you think of, or grapes have some of these antioxidants. So there are a lot of natural products have not been examined. The FDA doesn't require that these, some of these drugs are researched, or that they, because basically the natural products aren't regulated by the FDA. And since they're not regulated, drug companies can't get patents and they can't make money off of them. So even though they might have good health benefits, there's not a lot of research being done on those, because ultimately they can't make as much money. So just an interesting side here. So that actually concludes this part of chapter 20 that I wanted to talk about, kind of the overview of metabolism. All right.